I am really happy to be here hosting Alireza Mousavi to discuss his recent PLOS ONE Lab Protocol paper entitled Development of an Optimized Protocol for Generating Knockout Cancer Cell Lines Using CRISPR-Cas9 System with emphasis on transient transfection and the associated detailed protocol in protocols.io. Ali holds a master's degree in genetics from the Islamic Azad University in Iran. Since 2022, Ali has been approved as an elite researcher by the National Elite Foundation in Iran. During his undergraduate studies, Ali discovered his strong interest in cancer research and the development of new therapeutic approaches using cutting-edge technologies such as CRISPR. Building on his passion, Ali designed his master's research to focus on colorectal cancer, the third most common cancer worldwide. For his investigation, Ali developed a two-phase research plan. In the first place, his primary goal was to establish an optimized protocol for generating knockout cancer cell lines using the CRISPR-Cas9 system. This protocol aimed to enable the study of a wide range of gene functions in cancer cells. They successfully implemented this protocol and generated a variety of knockout cancer cell lines. Ali, thank you so much for talking to me today.
Thank you. Nice to meet you. And it's my pleasure to visit you here.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yes. Actually, our protocol is designed for generating knockout cancer cell line using the CRISPR-Cas9 technology with a focus on transient transfection using the lipid nanoparticles. And since it was essential for us to produce a knockout cancer cell line, we developed this protocol to be able to study these genes that play a critical role in cancer signaling pathway. Additionally, since we were studying multiple genes, ordering knockout cancer cell line from biotechnology company was not feasible option due to high cost and time constraint. By developing and optimizing this protocol, we create a cost-effective and efficient solution to generate high-quality knockout cancer cell line, making it highly relevant for advancing cancer research.
So I understand now the motivations for you to develop the protocol, but to understand its significance even further, can you please elaborate on what was the state of the art at the time that you decided to develop the protocol, to optimize the protocol? Were there any other protocols that existed to develop knockout cell lines and didn't work for you, for example?
Yes. At this time, it is necessary to say, although we accept many CRISPR protocols are available right now, but most of them are often expensive, resource intensive, and require reinforcing for a wide range of additional resources to complete certain process that omitted this main protocol. For example, I can some example of our process. Some examples such as essential step for performing MTT assay for calculating the best concentration of pyromycin, or designing the best guide RNA, or isolating single cell through the serial dilution are frequently omitted, that leaving researcher to search elsewhere for guidance. Actually, our main motivation to optimize this protocol was to create a user user-friendly, cost-effective, and comprehensive method that even researchers with a minimal expertise and resource could successfully implement. Actually, our main focus was on the developing country, because many researchers like us in developing country face significant financial constraint and lack access to the state-of-the-art facility and material. Also, they have often excellent idea and potential for breaking research, especially like cancer study. Due to the high cost of some technology, they are not affordable for the researcher to implement some study like CRISPR study.
As you were developing this protocol and moving away from the high costs of other protocols, was there anything that you tried to optimize that didn't work and didn't make it to the final version of the protocol? What were the lows and the highs of the process?
During the optimization process, we face a number of challenges in key steps, such as designing guide RNAs. As you may know, off-target effects remain a big solution and concern in the CRISPR study, and we adhere some strict criteria in order to reduce dramatically this phenomena. And for another method determining the optimal promycin concentration, because deviation of the optimized concentration may decrease dramatically the performance or efficiency of protocol. And isolation molecular cell population using limiting dilution is a very critical and tricky that require a high time and attention in order to optimize that. And on the other hand, developing this country coincided with the COVID-19 quarantine, which we faced with a significant challenge, particularly in obtaining material that we order from abroad. For instance, it took around 7 months to receive the initial material need to start the study. But overall, despite all of this difficulty, we preserved and ultimately developed a robust protocol that is compatible with a wide range of a cancer cell, ensuring adaptability and effectiveness for a diverse researcher need.
Ali, have you optimized the protocol further since publication? For example, have you tried to make this protocol work in cells in suspension?
Yeah. Before submission, we spent a significant time to optimizing the protocol to ensure it required no further adjustment. One of our primary goals was to develop a protocol suitable for a wide spectrum of the cell line. However, it's important to note that each cell line has a distinct characteristics for cell culture, and it's a researcher responsibility to adjust a condition as needed. For instance, in one sections that are related for calculating the optimal puromycin concentration, we emphasize that the suspension cell have more accessible surface for puromycin uptake, require a lower concentration in comparison with other cell for the adjustment. This flexibility allowed the researcher to adapt the protocol by ensuring a broad applicability.
I would like us to change gears now a little bit and talk about the motivations to send your protocol to PLOS ONE to be peer-reviewed as a standalone methodological paper. Why did you make that decision?
Yeah, actually, our motivation to submit this protocol to PLOS ONE as a standalone methodological paper stemmed from its focus on promoting transparency, reproducibility, and accessibility in science. PLOS ONE provide a reputable platform where detailed method can publish independently, ensuring they reach a broad and diverse audience. Actually, we believe our protocol addresses a significant gap in the field, in this field, and could serve as a valuable resources for a researcher. Publishing it as a standalone paper also allows others directly adapt, critique, or build upon methodology, contributing to the advance of open science.
Thank you. How did the feedback that you received from peer reviewers help you shape the final version of the protocol?
Actually, the feedback from the estimated peer reviewers was constructive and significantly made a change in our protocol. We address as many points as possible at the first version, which editor acknowledged us. The first revision was convincing and sufficient. There is some notable suggestion. One of that is some of the estimate reviewer suggestion why the Western blotting did not perform for validation. Also, we accept the Western blotting act as gold standard for evaluating the change of the protein level, but I want to say that we clarify this. I want to say that by sequencing of the target region, we validate the protocol performance and CRISPR function through the sequencing of the target region. Additionally, as I mentioned, they coincide with the quarantine, COVID-19 quarantine and string shipping policy. We face with a problem to receive from many material during the performing of this protocol, and it raise our cost, and it's not affordable for us in order to perform these tests such as Western blotting.
Why, Ali, did you decide to make the peer review history public together with your manuscript?
Yeah, actually, we decided to make the peer review history public because transparency is vital to fostering trust in scientific research. Much like blockchain technology, which ensured an open and verifiable record of transaction, making a peer review history public provide a transparent and immutable record of the review process. This approach allows the researcher to see evaluation of the manuscript from the initial submission to the final version, along with the constructive feedback and revision it's going. It assures the community that the researcher has thoroughly vetted, much like how blockchain validated and resources data. Additionally, this transparency promotes the collaboration and learning. Researchers, especially early career scientists, can gain insight into the peer review process and improve their own work. By making the review process open and verifiable, we contribute to culture, accountability, and trust in science, similar how blockchain fosters trust in the centralized system.
Thank you, Ali. The next one is my last question.
Yeah.
This, at this point, might be a little bit speculative because your protocol is relatively young. It was published at the beginning of November last year, so it is barely 2 months. But what has been, or what do you think will be the significance of this lab protocol paper for your young career as a researcher?
Yeah. Actually, development and publication this lab protocol have been pivotal for our career. It has showcased our ability to address the complex challenge in cancer research through innovative, cost-effective solution. These protocols emphasize on accessibility and reproducibility, has not only strengthened our technical expertise, but has also highlighted our commitment to advancing open science. Overall, publishing this protocol has expanded our visibility in the scientific community, opening up new opportunity for collaboration and demonstrating our capability to contribute impactful methodology. Overall, it has significantly enhanced our professional growth and reputation in the field of biology.
Thank you so much, Ali. I don't have any other questions. I don't know if you would like to add anything that hasn't been discussed yet.
Actually, I have no any question, but it is necessary to say that my supervisor and my teachers support me strongly at the end of this interview. I would like to greatly appreciate from the Dr. Arfa Mushiri, my supervision at this study, and also Dr. Hussein Fahimi, also my supervision and at this study, and also a special thanks of Dr. Nayer Ibrahim Zadeh for wholeheartedly collaboration for this study. Thank you all of my colleagues and teacher, because a strong helping of these people helped me to complete my research.
Thank you so much, Ali.
Thank you for your time and consideration.